Okay, so in this lecture, we're going to talk about how gluconeogenesis is regulated. So we're going to talk about gluconeogenesis regulation, and of course, we talked about the key steps of gluconeogenesis, so obviously those steps are going to be highly regulated. And we're going to talk about how the different enzymes are going to be turned on and off between fed and fasting stage. And the importance of gluconeogenesis, again, we'll highlight liver, gut, and kidney. So gluconeogenesis is going to occur during the fasting stage. It's going to be a major responsibility of your liver cells to maintain blood glucose levels. It also can be turned on during prolonged exercise. Under conditions of stress can also turn on gluconeogenesis. So you're going to have glycerol, which is going to become available because during the fasting stage or stress, your adipose tissue releases free fatty acids, and a byproduct of releasing free fatty acids is, of course, releasing glycerol. Lactate is going to be produced not only by red blood cells, but also by muscle cells doing exercise, or if it's a stressful condition, it could be an exercise, fight or flight type situation. Amino acids are going to be released whenever insulin is low, and then the amino acids can be used, of course, by your muscle cells for energy. Okay, so the regulation is going to be based on the activity of enzymes or the amount of enzymes present. The three key pathways are going to be, of course, pyruvate to phosphoenolpyruvate, fructose 1,6 bisphosphate to fructose 6-phosphate, and then, of course, glucose 6-phosphate back to glucose. So all of these are going to be tightly regulated. Now we have down here, we have our pyruvate, and pyruvate is going to become oxaloacetate, and this is turned on when there are high levels of acetyl-CoA. High levels of acetyl-CoA are, of course, going to be present because you're doing beta oxidation of fatty acids and generating a lot of acetyl-CoA. So that's going to turn on pyruvate carboxylase. So your pyruvate is going to become oxaloacetate, and then that's going to become phosphoenolpyruvate. Now, phosphoenolpyruvate will become pyruvate in certain situations as well. And so if you have a pyruvate kinase that's going to be active, then you're going to reverse that and go back to pyruvate. There's also going to be your other steps are going to be tightly regulated, which are going to be your fructose 1,6 bisphosphate and interchange between fructose 6-phosphate. And so remember when we talked about glycolysis, there was fructose 2,6 bisphosphate. And sort of as you had an increase of fructose 6-phosphate, remember that this became fructose 2,6 bisphosphate. And this is just simply saying that we have too much fructose 6-phosphate carbon. We have to push it to 2,6 bisphosphate, so that's going to be a positive regulator of phosphofructokinase 1 because it wants to make that fructose 6-phosphate into fructose 1,6 bisphosphate. So this does not happen during fasting. So during fasting, this is turned off. And so you don't have high levels of fructose 2,6 bisphosphate. You have low levels of that, and so you're not going to prevent fructose 1,6 bisphosphatase. And so your fructose 1,6 bisphosphate can now become fructose 6-phosphate. And then, of course, up on top here, you have your glucose 6-phosphatase and glucokinase. Glucokinase has a high Km, and if there's not a lot of glucose around, it's not going to convert it back to glucose 6-phosphate. So your glucose 6-phosphatase is on and can release glucose. All right, pyruvate is going to be key for gluconeogenesis, and keep in mind, pyruvate is, in the case of fasting, is going to be generated from mostly lactate and amino acids. It's not being generated from glycolysis. Pyruvate dehydrogenase is going to be inactive in the conditions of fasting where you have low insulin, high glucagon. It's also going to be phosphorylated to the inactive form, and acetyl-CoA levels, because of beta oxidation, is going to activate pyruvate carboxylase. So high levels of acetyl-CoA can activate pyruvate carboxylase and inactivate pyruvate dehydrogenase. Now, phosphoenolpyruvate carboxykinase is induced by cyclic AMP. And of course, cyclic AMP is going to be created following glucagon and epinephrine. Both of them will generate cyclic AMP. So cyclic AMP is going to activate protein kinase 4, which stimulates the phosphorylation of that Krebs factor, and that's going to go to the nucleus and stimulate transcription of phosphoenolpyruvate carboxykinase. Pyruvate kinase is inactive when phosphorylated due to high levels of glucagon. Now going to converting fructose 1,6 bisphosphate to fructose 6-phosphate. This is at that rate-limiting step of glycolysis, PFK1. Under gluconeogenesis conditions, the concentrations of the compounds that activated PFK1 are going to be very low. So fructose 2,6 bisphosphate levels will be low because we have very little fructose 1,6 biphosphate. AMP levels are going to be low, and so you're going to drive the on of fructose 1,6 bisphosphatase and off the PFK1. Okay, so now in your blood, you have glucose 6-phosphate is going to become glucose. Oops, 6. And this is going to be glucose. And keep in mind that glucokinase is going to be off. And that's because it has a high Km. So this is due to high Km. So it's not going to be active. And so you're going to be able to release glucose into the blood. Now, the synthesis of energy does require glucose. As I mentioned before, it takes ATP to reverse all of these reactions, and you don't form ATP. So the energy is going to be in reducing equivalents of NADH. And under fasting condition, of course, this all comes from beta oxidation of fatty acids. Now, sorry, this didn't come out too well again. I don't know what's happening with the conversions. But so as we've already talked about, after a high carbohydrate meal, glucose levels are going to rise from a fasting level of about 100 mg per deciliter. So this is fasting. To about 120 to 140 during fed. And this is glucose levels. And then, of course, you're going to return back to your 80 to 100 after about a couple hours after eating. If the levels were continue to rise, then water would be released from the tissues. So if glucose you get water release. And this is an osmotic effect. And this is going to eventually lead to a hyperosmolar coma. And so this is important, for example, in type 1 diabetics. Now, if levels were continued to drop, glucose levels, let's see if I can fit this. So this is 1, let's make this 2. If they continue to drop, then you get lightheadedness, dizziness. It can lead to a coma and you can get damage to your brain, etc. So maintaining blood glucose levels is going to be critical for just normal function of your body. So we've seen these pictures before. Just to reiterate, your glucose levels after a glucose meal, your glucose levels spike, insulin follows glucose, and then your glucagon levels will drop, but will stay at a certain range. Protein meal, again, your glucose doesn't spike, so insulin won't spike very much. It spikes a little bit, and glucagon levels actually increase during a protein meal. Oh, this got all flipped around. Sorry about that. So this is just talking about glucagon levels decreasing following a high carbohydrate meal, but they'll increase following a high protein meal. And then in a mixed meal, you're going to have both your carbs, your proteins, and fats. Your glucagon levels remain pretty constant, and insulin, because there is carbohydrates present, will increase during a mixed meal. So about 4 hours after a meal, you're going to have enough of a low glucose level, hormonal changes, so your insulin's down, your glucagon levels are back up, and you're going to start gluconeogenesis. You're also, by that time, going to also have the precursors for gluconeogenesis. Enzymes that are going to be part of gluconeogenesis are active under fasting conditions. So you have your pyruvate carboxylase, you have your phosphoenolpyruvate carboxykinase, your fructose 1,6 bisphosphatase, and your glucose 6-phosphatase are all going to be induced. And so the quantity of enzymes are going to be increased through those Krebs binding, your cyclic AMP response elements. Fructose 1,6 bisphosphatase is also active because levels of fructose 2,6 biphosphate, which is an inhibitor of that enzyme, are going to be low. So remember, fructose 2,6 biphosphate is an activator of PFK1. So if those levels are low, then also PFK1 is inactive. When you are exercising, you're going to have very similar methods or mechanisms as the fasting state, and this is again to maintain blood glucose level. And the liver is going to maintain these blood glucose levels through glucagon and epinephrine induced glycogenolysis and gluconeogenesis.